Hi everyone, so my name is Léonard Sauvé, and today I will present this paper, this DE-Kupl paper that was published in 2017 in Genome Biology Methods journal by Jérôme AUDOUX and colleagues from various institutes in France. They came up with this really interesting paper that I wanted to review for you guys so you can have a good idea of what they're proposing. And so the format of my presentation will be a readout of the paper in itself from page 1 to 11. So I won't comment the methods section, but I will comment every other section of the paper because I think it's very important to really go through this paper paragraph per paragraph so we can understand what they are saying, because there are new ideas in this paper, and I think we have to be very thorough when we analyze those results. So let's jump and start with the abstract of the paper. We introduce a K-mer-based computational protocol, DE-Kupl, for capturing local RNA variation in a set of RNA-seq libraries independently of a reference genome or transcriptome. DE-Kupl extracts all K-mers with differential abundance directly from the raw data files. This enables the retrieval of virtually all variation present in an RNA-seq dataset. This variation is subsequently assigned to biological events or entities, such as differential long non-coding RNAs, splice and polyadenylation variants, introns, repeats, editing, or mutation events, also exogenous RNA. So applying DE-Kupl to human RNA-seq data sets and identify multiple types of novel events and is reproducible across independent RNA-seq experiments. So this is the general idea of what they do, and we'll go through every step they describe in this paper. So as the background, they start by introducing gene code and sequencing technologies. So they say that successive generations of RNA sequencing technologies have bolstered the notion that organisms produce a highly diverse and adaptable set of RNA molecules. Modern transcript catalogs, such as GeneCode, now include hundreds of thousands of transcripts, reflecting pervasive transcription and widespread alternative RNA processing. However, despite years of high-throughput sequencing efforts and bioinformatics analysis, we contend that large amounts of transcriptomic information remain essentially disregarded. So, what do they say with this introduction is that we have now transcript catalogs that serve as reference to map our RNA-seq libraries. GeneCode is one of them. GeneCode is a very big compendium of transcriptomes that were gathered and then annotated, and we hope with GeneCode to capture virtually every transcript variant. But the others claim that this is not sufficient, and we'll see why. So first we have to describe the three major classes of biological events that drive transcript diversity. So first off, there is this transcription initiation that is thought to occur everywhere on the genome. So by that we mean that multiple alternative promoters exist at protein coding sites. Also, we also see that there are multiple alternative promoters for non-coding genes. And finally, we also have multiple antisense and intragenic loci that are transcribed. So there are three types of alternative transcription that is happening not only at the protein coding sites, but everywhere on the genome. So we assume that this is true. Secondly, large variety of mechanisms regulate RNA processing once the RNA is transcribed. We mean by that splicing events and also polyadenylation of the transcripts. We also have some RNA editing that can happen. And now we know that there are a couple of evidence showing that there is circularization of some RNA molecules and cleavage degradation by the nucleases. So they can be destroyed and then cleaved in some very various fashions. Thirdly, we have this genomic variation, so polymorphisms. How we inherit so different alleles. We have an allele from our father and mother, and so they generate polymorphisms in the population. So this is genomic variation that will be captured, of course, by RNA sequencing. So they call this single nucleotide variations. We also have those tandem duplications that are really proper to a person's transcriptome or genome. So duplications and deletions, transposition integration, so parts of the genome that move, unstable satellites or fusion events, and so on and so on. So those are all the polymorphisms that exist, and they are reflected in a RNA-seq library. So, and what happens with those three major classes of biological events? Well, current bioinformatics strategies, they do not capture them in their pipeline. So the others, they regroup three major strategies to analyze RNA-seq data. First, we have the first strategy of aligning or pseudo-aligning RNA-seq reads on a reference transcriptome. So this is the really state-of-the-art approach. And they mentioned a couple of papers that use this method. Is very state-of-the-art. Although it may be used in detecting isoform switching events, this analysis is by definition limited to transcripts present in the input reference. That's plain logic. Secondly, some methods will try to reconstruct full-length transcripts based on read, based on fragments. This is not usual, but it exists, and some people have shown that it could be used and could be good in certain instances. But although these protocols can identify novel transcripts, they do not account for true transcriptional diversity, as they ignore small-scale variations. So they try to reconstruct full-length transcripts, but if you have only evidence of half of it, or for smaller events, then they will ignore. Thirdly, we have the discover of specific events. And this task is done by multiple various programs that each are specialized in discovering specific events from libraries. So, and finally, strategies that combine multiple software items for a comprehensive transcriptome analysis are difficult to implement and cannot be truly exhaustive. So, why DE-Kupl? Well, DE-Kupl tries to, so in this paper, they will try to demonstrate two things. First, that large amounts of captured RNA variation is not represented in existing transcript catalogs. So this is their first task will be to show that. Then their second task will be to propose a new approach to RNA-seq analysis that facilitates the discovery of the transcript variation events. And they will try to follow 3 key points: that it will be independent of transcript, assembly, or alignment. So they won't be trying to assemble the reads into transcript, full transcripts, or align them on a genome. Also, they want their approach to be k-mer-based, and we will discuss what k-mers and what are those implications. Finally, they want to identify biologically meaningful transcript variations. So, following those 3 points, they will propose a new approach that is called DE-Kupl, and DE-Kupl means differential expression analysis of k-mer content. So for their first part, they want to prove or show that reference data sets are incomplete. They do not fully present the diversity of actual transcriptomes. And they show it by with this first figure. So the figure 1 of the paper is this one. And so what they do is they show the measures of k-mer abundance in each of those 3 categories in the genome, in the gene code. So the gene code database, transcript catalog, then the human reference genome parsed into k-mers. So the k-mer indexing is done on this genome of reference, and by each of their RNA-seq libraries. And what they show is that first, they noticed that most of their k-mer coming from their RNA-seq libraries, they, for the most part, map to this gene code catalog. So this is pretty much expected in abundance. However, when they consider k-mer diversity, so the copies on the unique k-mers, then the tendency is quite opposite. So while most map on the gene code, there's a large proportion that maps only on the reference genome, however. For example, with this bone marrow RNA-seq library. So indicating that many k-mers weren't assessed, weren't there in the gene code catalog. So, and they assume that these tissue-specific k-mers may result from sequencing errors. For example, if we take the bone marrow, so 14% of the whole content of k-mers is unique to bone marrow in their experiments. So this is library specific, and they can arise from sequencing errors, genetic variation, or maybe novel or non-reference transcripts that are not annotated in the gene code. Then they also notice that the majority of the RNA-seq k-mers that do not occur in gene code are found in the reference. So they are found in the reference genome. That wasn't in the gene code, but it was on the reference. So this is a very clear representation of what they want to say is, well, most of our stuff is mapped on the reference genome. Why doesn't it appear in the gene code database? That is quite troublesome. And they want to answer that. So further scrutiny of tissue-specific k-mers show that many can be mapped to the transcriptome with one substitution. So what they show here is that if we're not able to map on either gene code or the reference genome. Then with one mismatch, then it's okay. It's annotated in the gene code. With two mismatches, yes, some will map on the gene code, and the rest will be plain unmapped. It's too much errors, too library specific, and we cannot do anything with that, with those schemas. But furthermore, what they try to say is that those non-reference schemas that don't appear in gene code nor the reference, well, those they contain some information. So what they do is a principal complement analysis of the human tissue samples using conventional transcript counts and chimer counts. So these are conventional transcript counts, and this is the chimers that they sampled that are very specific to the tissue. And we see that in all of those cases, PCA is able to really cluster the different tissue types at their corners, and basically they want to say that this approach yields the same result as this approach. Interesting to see that there are two groups here, but it's not very important. It's that those three groups, they cluster very at different parts with chimers that are not referenced in any of the catalogs. So this illustrates that biological relevance of non-reference transcriptome is not accounted in standard analysis. So this doesn't appear in any current bioinformatics analysis so far, but they say that it contains truly information about tissue, for example. And finally, what they show is that library-specific chimers are observed much more frequently in RNA-seq data than in WGS chimers. So WGS is whole genome sequencing, is just DNA sequencing. So, and this is interesting because they perform whole genome sequencing and gather chimers and split them into chimers. Then they use the same sample to do RNA-seq libraries, and they just look at chimer diversity. So how much chimers they have in common. And in what we see is that while most of the chimers coming from this WGS are also the same as the one in the reference genome. So this is pretty much expected from RNA-seq data, sorry. But we should expect this same proportion in RNA-seq data, but it's not the case. We see that almost half of them, of the chimers originating from the RNA-seq data, they just don't map on anything. And neither gene code, the reference genome, nor the whole genome sequencing. So many of this is very RNA-seq specific. And this we want to retrieve and to analyze with some more precise methods. So maybe with chimers, we will be able to really know what's happening in there. So in summary for this results section, we see that reference data sets are an incomplete representation of actual transcriptomes. Why? Well, when we consider chimer diversity, a large proportion of chimers are tissue specific and not found in the gene code reference. Secondly, these tissue specific chimers may result from 1, sequencing errors, 2, genetic variations in individuals, or 3, novel or non-reference transcripts. So altogether, these results suggest that the existence of a significant amount of untapped biological information in RNA-seq data. Now, they the authors will propose a new k-mer-based protocol for deriving transcriptome variation from RNA-seq data, with the aim of capturing all k-mer variation in an input set of RNA-seq libraries. This protocol has four main components that they describe as indexing, filtering and masking, then differential expression, and extending and annotating those differentially expressed k-mers. So, quickly going through the figure that describes this approach, we see all the different libraries here in FASTQ format. They then will be indexed using jellyfish and gathered into a k-mer count matrix with layout library 1, 2, 3, up to n. And each of those colors is a k-mer, a different k-mer. Then one important task that will be done is filtering and masking. So they will apply some filters and delete some of the k-mers based on some criteria that will be discussed later, so that this count matrix is reduced in size. Then they will compare those abundances of k-mers with differential expression analysis. Similar to regular differential expression analysis for gene expression, they do it for k-mer expression. Of course, between condition A and B, could be anything, could be male, female, could be treated, untreated. Yeah, so any condition, they separate the two groups and then apply some statistical test to see if those k-mers are differentially expressed. Then the ones that will be selected by the differential expression will be, we will try to assemble them into countigs. And after that, they will map those countigs to real biological events. So you may have noticed that regular approaches for RNA-seq analysis, they do this as the first step, or in the first steps of the, so they had many different pipelines exist, but this step is usually done at the beginning, because you filter out some of the reads that cannot be assembled together, and you kind of get this filtering from contig assembly. But they do it at the end, and they do it at the end because they are working with smaller fragments with k-mer counts instead of with k-mers instead of reads, so RNA fragments. Some accommodations that they do for, so indexing, filtering, masking, and so the upcoming steps. First, they have to accommodate for the large size of the k-mer index. A single human RNA-seq library contains of the order of 10 to the 7 to 10 to the 8 distinct k-mers. So this is quite large. It's a large number of k-mers that we produce as humans. So they will select the jellyfish tool for counting k-mers. So they won't be optimizing this part. So this indexing, they will use a current tool for that, which is called jellyfish. Very fast computing times, and allows the storage of the index on disk, which is nice. So for what is really the contribution in their method is this. So masking and filtering, what they do as pre-processing step, so post-processing steps after k-mer indexing is very important, because it allows them to reduce the size of the k-mer tables they're dealing with. So they will filter out unique or rare k-mers. that is quite straightforward and very ubiquitous in the field. So k-mers that are really low number, they will rule out, they will just mask them out. Also, so filtering is just filtering bad k-mers, error k-mers. So this is very straightforward. Masking is different. It's another type of filtering. And this is very, so I will read this to you and maybe you will understand that this is strange. So masking entails the removal of k-mers matching a reference transcript collection. So basically they will remove transcripts, they will remove k-mers that match perfectly some transcript collection. The rationale for this is that the bulk of k-mers in RNA-seq data comes from known exons, a form of canonical exon expression ignored in this study, as it can be captured efficiently by conventional reference-based protocols. So they will delete k-mers that map to exons, just because they want to look at more carefully into so not the exon expression, but all of the non-coding, the not very well annotated rest of transcripts that are expressed, but that we don't have a really good catalog for them. So basically, each k-mer that maps perfectly to an exon will be removed. To an exon annotated, of course, by either gene code or a simpler transcriptome or skipped altogether. So it's nice of the others to leave us the choice of using anything for masking, gene code or your own transcript catalog or skipped. This is cool. Also, some further accommodations would be for the differential expression analysis. They allow two modes, so the t-test mode or the DSEC2 base mode. So this one is used even for, so the two tests can be used in a regular gene expression, differential expression for gene expression. So they're state-of-the-art. This one is very fast and this one is slower but more sensitive. And also they recommend to use it for small samples. Well, and and finally, the k-mer extension procedure merge overlapping k-mers into contigs. And it will stop as soon as a fork is encountered. So this is some accommodation that they make. So when it's multi-mapping or there is ambiguity, then you stop merging to contigs. So there will be some redundancy in the contigs, but yeah, they prefer to just stop when a fork is encountered. And also they don't want to make full-length transcripts. It's just a procedure to group k-mers that represent the same biological events. Whenever possible, and this is really a strange appearance in the article. So whenever possible, the key steps of the procedure, k-mer table merging, t-test, and k-mer extension were written in C, enabling the whole procedure to run on a relatively standard computer in a reasonable amount of time. So this is funny to me because they really, so they insist on the fact that this part is long. K-mer table merging, t-test, and k-mer extension is a long process, and they say that they try to accommodate and make it fast, but still we'll see in the benchmarking results that it's very strange to to have extra steps that take so long to process, and so they will justify that it's very useful to merge and then map and then assemble, and that all of those steps are really critical to make the whole analysis. But actually they just take time, and yeah, is it really good to make all of those steps be in between? We'll see. Well, so those benchmarking results. So discovery of differential RNA contigs with DE-Kupl, with the aim of assessing DE-Kupl's capacity to discover novel differential events. So in this section, they will describe how their algorithm behaves on real data. The data is epithelial, mesenchymal, transition. So it's called EMT. The methodology they use is to apply DE-Kupl to 12 RNA-seq samples from an EMT cell line model, in which non-small cell lung cancer cells were induced by ZEB1 expression over a 7-day time course. Then they compare 6 RNA-seq libraries from the epithelial stage of the time course with 6 libraries from the mesenchymal stage. So they will compare 6 samples from one group with 6 samples from another group, and see what has changed, or what is used differently, so has a different usage. It's completed in about 4 hours in t-test mode here, and 6.5 hours in DSEC2 mode, even with multi-threading. It will use 8 computing cores and 55 gigabytes of RAM. So pretty extensive in terms of memory, and 7 to 42 gigabytes of hard disk space. So it depends on the index that you will keep on disk. Recurrence filters efficiently reduce k-mer counts from 707 to 92.5 millions. Entities, and gene code masking further reduced count to 40.3 millions, as we see on this figure. So they reduce the number of k-mers or contigs by applying those filters. So we see that those sizes, they're reduced. They're reduced extensively during those filtering and masking processes. This is nice because a 728 megabytes file is easier to manipulate than a 2 gigabyte file, or this one I can't even imagine how heavy it must be. So this is nice to have a much lightweight matrix. Also, they will identify 3.8 million differentially expressed k-mers, and they will merge those 3.8 million k-mers into 133,000 contigs assembled after assembly. So we start with this count of k-mers, and we go to this number of contigs that represent really biological events. They are contigs that are not exons. They are they're not sequencing errors for the most part, and they will encompass some biological events that happen, so transcriptional events. So they're very valuable contigs in the end. So the resulting contigs range from 31 base pairs to 3.6 thousand base pair. So this is the length of so the contigs. I'm sorry. So this appears, so it doesn't appear in the tables on the figure. So yeah, I'm sorry, here. So this is the content density per contig length. So, and they will observe this two peak thing on the density plots. And this is strange because the 31 base pair contig length is just the orphaned, unexpanded k-mer. So this is a k-mer that wasn't successfully assembled into a contig. So there are a couple here. Now the 61, how could this arise in analysis? Well, they had some idea. They thought that those 61 base pair contigs result from the assembly of 31 overlapping k-mers harboring a SNP at every position of the k-mer. This is a very strong hypothesis, but they will prove it by looking at the mismatch ratio. So they see that so when your contig is small, then you probably don't have mismatches on the reference genome. But as you go up in length and go to 61, then you see that this is your peak of mismatch ratio. So they say that this one peak represents like an artifact. It's just an artifact of the k-mer assembly into contigs of length 31. So when they assembled with an error, they map with, so they assemble to a single 61 contig, and that's why there are more 61 contigs in that context, because they fit into a really specific artifact set arising from the size of the k-mer hash. As expected, shorter mapped contigs tend to map at multiple loci, more often than longer ones. So we see here that shorter contigs map more often to multiple places than longer ones. So these are really conventional observations about contig mapping on reference genome. And also we see that, and this is very big, it's, however, 80% of all contigs are uniquely mapped. So 80% of all the loci of all the contigs that we identified here, they map to so 99% of that map to the human genome. So this is really good. This is really a nice result. And that among them, 80% were uniquely mapped. So it's a very rich and very good set of contigs that we know are uniquely mapped, so they're not unambiguous in the genome. And there are a really big number of them, and only a small portion is ambiguously mapped on the genome. Then, now they try to analyze those mapping results. So where are those contigs appearing on the genome? And so they will make the discovery of differential RNA contigs, and they will analyze the contig locations, and they will also reveal distinct contig classes. These are the 5 that will be discussed here. And they will they will observe firstly that the contigs are mostly annotated intron and exons. So here, intronic contigs are predominantly exact matches. Intronic are exact matches mostly, and exonic contigs are predominantly mismatched in exons. So this blue portion here. So you see that it's really the reverse here. we map to more introns than exons. And when we map to exons, it's in general more mismatched. Why? This is due to the reference transcript masking. Contigs with exact matches to introns are not masked because they are not in the reference, while contigs that match exons are filtered out unless they differ from the reference. This difference might be in the form of SNPs through exon extending into flanking or intergenic or intra-niche regions. Yeah. And by the same rationale, contigs mapping to intergenic and antisense regions are depleted. So they are depleted in total. This is normal because intergenic, they're very long and not really known regions. We don't have much information about them. Antisense are just very rare in the genome. So they're depleted, consistent with their location in unannotated inRNA and antisense RNAs. While contigs overlapping exon-exon junctions behave like exonic contigs. So they have this same proportion because they are junction between two exons. If they were perfectly matched, so if a chimer was perfectly matched into a junction, something that is annotated as a junction, it might be masked and removed. So this is why we have also a big mismatch portion of junctions. And finally, what I find the most interesting in those results is that you see that this big green portion here, so it's a significant fraction of exon junction contigs that are exact matches. So basically, we identified with this method novel junctions that weren't annotated in any transcript catalog. And so in there, we must have new junctions that weren't in the reference. And so this is a cool contribution, because now we have a means to identify novel junctions of exons simply from chimer indexing, filtering, masking, differential expression, and then mapping. Now, in the last part of their the analysis of their approach, they will spend some time explaining how the assignation of contigs to biological events work and how they came up with some rules for assigning what is a biological event happening from the RNA-seq libraries. So they generate this table with all the rules, and we will go through all those events and how they kind of describe. So this is the descriptor of how the mapping works, and this is the number of contigs they will identify with on which unique loci they mapped. Since intragenic differentially expressed contigs may result from a mere over or under expression of their host gene, they do not necessarily reflect a differential usage. So differential usage is DU of transcript isoforms. So they implemented a simple strategy to distinguish between the two situations based on the expression level of the host gene. So this will be described in the methods section that I won't discuss in this review. So they made this distinction for splicing, polydeniation, SNVs, and intron retention. So they're just basically explaining how this DU works. So they apply a DU, so a condition on the probability of one group having this particular k-mer or contig overexpressed versus the other. This is this p-value, and they will put a threshold on this p-value, and you have to be lower than 0.01 to be a splicing DU, not just a splicing event. Then, from the total set of 133 KD contigs, we extracted about 76 contigs matching our rule set for either event class. So this is a result. So they extracted from this almost half of it corresponds to a real event class. Note that certain events generate multiple contigs. We thus further grouped contigs into loci, and we describe below the main classes of events they identify. So they used some kind of grouping, so we have a good idea of what they are seeing with those results. So the first event we will see is the differential splicing event. And what they did is, so we report first the number of contigs that in the set, the 133K contigs. So this number was, this number of contigs were assigned with this splicing event. Regrouped on 1200 loci difference. So that means that some contigs map to the same loci. This is expected. Also, they identified that 391 contigs were differentially used between the conditions that they looked. That means that in total, 345 loci or even genes, we can now call them different genes, different splicing that were identified in those different conditions. So we see, first of all, that this number of different splicing, differentially spliced genes based on RNA-seq libraries, you wouldn't be able to get that from a regular analysis pipeline. You would need some specialized tool to perform this task. It's not, we cannot do that with regular gene expression analysis because those alternative splicing require really specialized tools. And they show it here on the figure is that, so we see that in condition E, so this is M. So M is mesenchymal and E is epithelial. So they compare the two conditions, and red is more expressed than M in that case, in that particular locus. And gene code has this annotated as a gene or some kind of splicing site. And we see that this region is expressed as well. So DE-Kupl noticed that and said that we can have this splicing or this truncated one that goes further. So basically, DE-Kupl identified a novel splicing event, and also that this novel splicing event is differentially expressed in those two conditions. And finally, they show the same thing with some other gene, or this is, I think, a tandem repeat. C is, no, this is a long intergenic non-coding RNA overexpressed. So this is a new event that was identified with decoupled, and they even don't show gene code because it won't appear in gene code. Now, assigning contigs to differential polyadenylation. So this is the result that we see here. And we see that there are a few less. And this is the rule sets that they use. So you have to be clipped at the 3' greater than 5. You have to map to a gene. You have to, so, and they have also another rule set that I won't discuss here. But so, and what they mean by a clipping, 3' clipping is containing 5 or more threading aids. Out of the 130 polyterminate contigs, only 105 contain an AATAAA or variant polyadenylation signal. So basically, it's always this number you want to look at is the number of differentially used polyadenylation sites. 8. So we have 8 novel adenylation sites that were differentially expressed in the 2 conditions. This is cool. This is still new results that you wouldn't get with state-of-the-art results. Then, for long intergenic non-coding RNAs, they identify a subset of 1061 differentially expressed contigs on 329 loci. So this is the result C that I showed you before. And you have to be so greater than 200 to be to consider the long non-coding intergenic. Yeah, we can cluster into well-defined transcription units with abundant read coverage and evidence of splicing. So it's kind of yeah, you see the pattern here. It's DE-Kupl identified this as a as a link RNA because there are evidence of multiple splicing events. It's long and between 2 genes, and it's differentially expressed. So all of those criteria are respected. And of course, you can have a tool that does it, but DE-Kupl does it as well, among other things. For antisense RNAs, then you have to not map a gene, because you're an antisense RNA, so you're mapping to the exact opposite strand of the gene. You have to map antisense genes, true. You have to be mapped, yes. And then the contig length has to be greater than 200, because if not, you're not really a gene, you're nothing. And we see here in figure D is that we have an antisense overexpressed in the mesenchymal state of this particular gene. So the gene is going in that direction, and we have this expressed antisense gene here. It's not really clear. They say it's a novel antisense RNA. RNA secretes RNA in the forward orientation, while the gene at this locus is in the reverse orientation. The annotated gene is not expressed. So yeah, this is what I wanted to verify, is that, yeah, they see that it's not expressed in either conditions. And they say that when DE-Kupl is applied to stranded RNA-seq libraries, the resulting contigs are strand specific, and thus we can use that to identify antisense RNAs. So they identify 479 contigs originating from 180 loci, mapping to the reverse strand of annotated genes. So these antisense RNAs, very strong cases of differential expression, sometimes combined with apparent repression of the sense gene. So this is interesting. That's basically saying that if this antisense gene is expressed, then you will have repression of the normal sense gene. Now, assigning contigs to allele-specific expression is more complicated, but they can do it, they attempt to do it. So, DE-Kupl quantified every SNV containing K-mer. So, they try to exploit this capacity to identify potential allele-specific expression events. And they extract contigs including SNV. So it can be a base substitution or an insertion deletion, and for which DU was predicted. So you have to be differentially expressed, you have to map to a gene, you have to have SNV, be exonic, you map to at least one region, and also another set of rules. So in that sense, they identify now 680 loci that display strong apparent changes in allelic balance between the epithelial and mesenchymal conditions. So the ability of DE-Kupl to capture differential SNV between data sets might be particularly relevant when looking for recurrent mutations in subpopulations. So once you know that the SNV is particularly relevant, then you can focus your analysis on this SNV in different conditions. This could be very interesting, and this also can be done with other tools, but it still can be done altogether with DE-Kupl. Then we have so the assignation of contigs to intron retention or other intronic events. As highly expressed transcripts often carry intronic byproducts, we expect DE-Kupl to identify many parasitic intronic contigs. And this is what they try to extract. And indeed, there are almost 50,000 contigs that are mapped to intronic loci. This is very expected. We thus focus on intronic K-mers for which DU was predicted. So this value here, and we see that, so 3100 K-mers, so loci, sorry, will be differentially used and originate from intronic regions. And this is, I repeat, this is cool because we can do it with annotations, but with that particular approach, we completely bypass annotation of the intronic regions, and we just base our analysis on K-mer abundance from the raw files. And then applying some filters. So these ones are very curated, very precise loci that we know are differentially expressed and are not genes, that are not annotated as protein coding genes or anything else. They are intronic regions. Finally, we have the expressed repeats results, and this is very tough to assess. Is so because so as ambiguous alignments render repeat regions unmappable. It's a very old problem in bioinformatics. Repeats are very hard to map, and so gene code will annotate a region to be a tandem repeat, but with DE-Kupl, you'll see that all of those black spots are contigs, so we see really clearly with DE-Kupl that this is a really repetitive region, and so DE-Kupl retrieves that information as well. And also in the same way, it will retrieve the same sequences as other tools as well. So this is not really important because this is a very refined problem, a very old problem of finding repeats, but DE-Kupl is attempting to do the same with more or less success. But yeah, and there is some proportion of contigs that won't be mapped to anything, won't be mapped to the reference, or wasn't removed during filtering or masking. So those transcripts, where do they come from? Well, they come from for the expression vector used for inducing the cells, for example, or non-human primate sequences. So they identify that those contigs are just pollution or were integrated in the genome some way. They map to nothing, but they are very small. This is a very small proportion. So this is basically the whole table, the whole sets of rules for genetic events. They really classify them, and with thorough investigation of those of the DE-Kupl report, we can safely associate K-mer, and so directly from the K-mer tables, some biological events. Now, the authors will discuss the impact of transcriptome masking in their results. So, since they are using GCOL as a reference transcriptome, they analyze the impact of using different reference transcriptomes on their results. So they wanted to show what is the difference between lightweight masking and the absence of masking. With the lightweight transcriptome masking, they noticed a very moderate impact on the number of differently expressed K-mers and contigs. However, when you just skip altogether this masking, then suddenly a large increase in differentially expressed K-mers and contigs arise. It's going from 3.5, so it's a 3.5 and 2.5 fold difference. So it's very big. We recuperate much more differentially expressed K-mers by not doing this masking. Importantly, less stringent masking produced longer contigs because you can assemble contigs more easily because there are more, and a higher number of detected events. So you gain some sensitivity by not masking, by not doing this. So by less stringent masking procedures yield an increased number of events. So I'm quite unsure about this. So this result, because this is saying to me we should skip masking altogether. We should not remove any K-mers before doing this differential expression analysis. This is saying that to me. But the thing is that lightweight masking may be the preferred option because it's returning a higher number of events for a little additional computational cost. If you have your computational cost being not a problem, then do no masking at all. It should be more precise. Benchmarking results about the discovery of differential RNA contigs. They want to further assess the power of their method on other, so compared to other tools and compared to other data sets. And this is a very crucial part. When you present another tool, you want to see the performance against other methods. And so comparison with specialized tools, I won't show the results because they won't show it in the main section of the paper. I assume that it wasn't very important for them to show that. And I trust that DE-Kupl is able to do as well as other specialized tools for polyadenylation, circularization, and so on and so forth. They are basically the results for identifying novel biological entities that were described in table 3. It can be performed with other tools, but DE-Kupl does it all together in one analysis, and it can do even more. So there is no real comparison to make with other tools because it's completely novel. But we can compare between data sets. So they did all their experiments on 12 RNA-seq libraries. Now we want to see if they can expand those results to much larger data sets. Not larger data sets, but other types of RNA-seq libraries. And so they took the GTEC and the HPA data sets to test. And what was their methodology is to identify differentially expressed contigs using DE-Kupl on 8 colon versus 8 skin library from GTEC. It's 8 versus 8 in GTEC using DE-Kupl, then identify the differentially expressed contigs, classify them into events, and the top 100 top events from each class were extracted and their Kmer labels saved as a sequence file. When then counted we then counted the occurrence of each Kmer in the colon and skin libraries from the HPA project and applied DE-Sec2 to evaluate the significance of the expression change between colon and skin. It's complicated. It's not the best way of benchmarking if you want. So to me, it's not the best way. But what they did is simply try to compare differential expressions between those two data sets. So GTEC events, unmapped, splice, repeat, poly, link RNA, or intron. These are all the P values. And they say that in the protein atlas, it's most of the time also in the range of significance, the same significance. So they say that those that were identified in the protein atlas that are called as will call as unmapped is also the ones that were identified as unmapped in GTEC. So they say that those two annotations or biological entities calling, they are the same in those two, so they are similar or comparable between those data sets. Altogether, 79% of the 550 differential expressed Kmers identified by GTEC were also significantly differentially expressed in the HPA data. So it's a very messy way of comparing results, but with differential expressions, since we don't have the ground truth, we don't know if those genes are really differentially expressed in either conditions. Plus, we're dealing with unannotated events. It's very hard to compare if those callings are similar or comparable. So this is my main critique about this benchmarking. It's very hard to understand this figure. I encourage that you take time to just understand the intricacies of comparing those p-values and seeing that everything matches up, but it's a fair demonstration that those results are comparable. But it's a hard task to assess if it's very exportable to other data sets in that setup. Now, I will comment some of the discussion notes that the authors make about their research. First, in contrast to popular RNA-seq analysis software, DE-Kupl does not attempt full-length transcript assignment or assembly, but focuses on local transcript variations instead. So first note, it's different from other methods. Second, they do not consider full-length transcript. To be realistic when screening for unspecified RNA variation, since the combinatorial nature of genomic, transcriptomic, and post-transcriptomic events would require an indefinitely expanding transcript catalog. So they are saying, well, it's no use to try to catalog every transcript. We won't be able to do that. Even gene code is extensive, but it's not extensive enough. Don't try to do that. It's not useful. It's not the way we should go at it. In this sense, DE-Kupl is closer in spirit to other methods analyzing local RNAs coverage. So they want to do the parallel of their method with previously proposed methods. It's completely novel, but it's similar in spirit to other methods that don't try to bypass reference and to really pinpoint the local variability. Another important benefit of the k-mer strategy is that the k-mers representing events of interest can be used efficiently to assess the occurrence of similar events in the huge public compendium of RNA-seq data. I think this comment is not useful in a paper because this is true for anything. So anything you find in your data set, you should find on other data sets, other compendiums. It's true but not useful to say that. In this proof of concept study, we analyzed RNA-seq libraries from a small number of individuals and from a single cell line. We expect k-mer diversity to rise significantly with the number of individuals included in the analysis. And this, they will explain it. I find it really funny because they do an experiment that shows that, so they, however, preliminary tests with over 100 libraries from the Cancer Genome Atlas show a sublinear growth. So I would like to see that. It's in the additional file, so it's very deep in the paper, but the data is here. I would have liked to have this result before because scalability is very important in those kinds of analysis. 8 versus 8 is quite small. It's a small number of data sets that we analyze. So with 100 libraries, it would be interesting to see if it scales. So it suggests that there is good scalability for DE-Kupl. I'm not convinced, but we have to make those experiments happen and test with DE-Kupl and see if we can scale up. Analysis of large-scale patient RNA-seq data opens exciting perspectives. That's true. For instance, the ability of DE-Kupl to detect genetic variation and RNA expression processing events simultaneously may serve as a basis for studying genotype-phenotype relations. Yes, among other things. Analysis of patient RNA-seq data may also reveal event classes not studied in this work, such as fusions transcripts and circular RNAs. So they're basically saying that we should maybe extend the decoupled procedures to have to detect other biological events. But the framework is there. We just have to make those rules to really make to make the mapping of a contig to a novel non-coding RNA event or transcript biological event. In conclusion, there are some key aspects of this work that are really good to have in mind after going through this research. So exhaustive capture. So DE-Kupl is an exhaustive capture of biological variation in IRIC data through k-mer decomposition. K-mer decomposition followed by filtering, masking, and DE analysis is a novel way of analyzing RNA-seq data. It can detect a wider spectrum of transcript variation in previous protocols. DE-Kupl explores all k-mers in the input RNA-seq files versus only k-mers from annotated transcripts in recent software analysis. So they really go deeper, which potentially requires substantial computational time and memory resources. And this is discussed in the paper. Using the Jellyfish k-mer indexing software and C program code for the key table manipulation, they achieve time and memory requirements on par with popular mapping-based software for similarly sized datasets. That's true. So 6.5 hours of computing time for 12 libraries. It's quite similar to what you would get from aligning with STAR, then doing a DSEQ2 analysis. So any conventional approach would take the same amount of time. A key aspect of our protocol that rendered a full k-mer analysis tractable with the application of successive filters for k-mers, reference transcripts, and differential expression, which altogether resulted in a 200-fold reduction in k-mer counts. So I don't know if they are proud of that or not, but they discussed the implications of filtering and masking in their procedure, and they say that it's good because it reduces the size of the table. I agree, but it's more a fact. You have to take that into consideration. When you use DE-Kupl, there will be filtering, there will be masking, and you have to control those parameters and be able to assess the implications of those masking and filterings on your analysis. These filters are not only useful for technical considerations, because they reduce runtimes and enables to get rid of most sequence errors, so this is good. But also they allow the user to focus on k-mers that want vary significantly between the conditions. That's a good point. Your analysis will be more precise if you get rid of the unnecessary variations that you don't want to assess. So yes, focusing, so filtering and masking for very significant k-mers that are differentially expressed is good. It is good in a way. And secondly, it's good because it encompasses events that would not be captured by conventional reference-based protocols. So this is very key to understand the method. The k-mers, by doing this k-mer approach, we tackle a part of the genome that is not captured by the reference protocols. So this is a very a very huge step up in RNA transcription analysis. And plus that we can identify a large variety of transcription events is even better, because we now use one program, one pipeline to really safely pinpoint the loci that are transcription events that are not annotated and that are relevant. We have strong beliefs that they are relevant in the transcription landscape of the cells, different conditions. And finally, so they show that DE-Kupl is able to detect a wide range. So much what I was saying before, a wide range of differential transcription and RNA processing events. Although specialized software may perform better at assessing specific event classes, such as differential splicing, no method known to the others provides such a comprehensive screen. This is true. As differential RNA-seq analysis is often conducted with an exploratory spirit, we argue that it is preferable to cast a wide net with no preconception for target events, using DE-Kupl along with a conventional gene-by-gene differential expression analysis. Note that DE-Kupl might also be an interesting option for exploring other types of next-gen sequencing data, such as small RNA-seq, ChIP-seq, or whole genome or exome sequencing. So, and they say after adjusting its parameters and event annotation rules. So they're opening up to the fact that we should contribute to making DE-Kupl always able to do more. But we have to be very careful about the parameters and the adjustments we have to make to the program. And also that they insist on using this approach along with a conventional gene-by-gene differential expression analysis, which is a big takeaway from this method. You saw that there are a couple of filtering, masking, counting, we're assembling into countings, and so complicated steps to get around this annotation gap that we don't have, that we do not possess. We do not fully understand transcription variability, and DE-Kupl is there to help us understand this diversity in transcripts. But we still have to rely on previous methods of really state-of-the-art methods to assess the differential expression for genes, for protein coding genes, for the ones that we know have impact, that we have data on, that we have studied in the labs. Those genes, we have information, and we know they affect, so they are affected by different conditions. And so for this, conventional analysis should be prioritized at any time. But for this really black box of transcripts that exist and that we don't know how they function, we should have more precise protocols to do that. And DE-Kupl seems to me like a very suited protocol pipeline for analysis of RNA transcripts. So the whole story, to get a full picture of the RNA diversity in different conditions. Thank you for your time, and I hope this was instructive and will incite you to use DE-Kupl for your gene expression analysis in the future. Thank you.